The overall goal of this video is to provide a detailed description for the GC-based analysis of the aldononitrile acetate derivatives of glucosamine and muramic acid extracted from soils. The process begins with acid digestion, breaking down organic polymers and releasing glucosamine and muramic acid monomers. Amino sugars are then isolated and purified from the mixture solution. This is followed by derivatization, to convert those amino sugars to volatile aldononitrile acetates for gas chromatography-based analysis. The final step of the procedure is the liquid-liquid extraction and GC determination. Ultimately, results can be obtained that show baseline separation of the aldononitrile acetate derivatized amino sugars, eluting between the myo-inositol and methylglucamine standards, through GC with flame ionization detection.
The main advantage of this technique over other methods is that it optimally balances precision, sensitivity, simplicity, good chromatographic separation, and stability upon sample storage.
This method can help answer key questions in the field of global carbon cycling, such as the contribution of senesced microorganisms to pools of stable soil carbon. Although this method can provide insight and information into the accumulation of microbial detritus in soils, it can also be applied in other matrices such as ocean systems, biological tissues, and even extraterrestrial samples.
To begin this protocol, freeze-dry soil samples after field collection.